Good morning, ladies and gentlemen. Special welcome to the family. To this ceremony, which I hereby open, and in which Dr. Gabriele Addario will defend the academic thesis entitled Biofabrication Platforms, Advanced 3D Bioprinted Models for the Study of Kidney Fibrosis. Before we go to the opposition and the defense of your thesis, I would like to invite you to present your major findings and conclusions of your work in a short presentation. Please go ahead.
Thank you. Okay, so I will give actually a brief overview about the work that we have done in these last years. So my PhD thesis has been developed on different chapters. In the first chapters, we actually started from reviewing multiple papers and publications with the aim to actually understand more about kidney fibrosis and also kidney models. Then from chapter 4 onwards, we actually started to work experimentally in the lab, and our aim was to first develop two different proof of concept in chapter 4 and in chapter 5, where in the first chapter we actually use a bioprinting technique, while in the second one a printing technique. And our aim was to really try to mimic certain aspects of the kidney architecture. Then in chapter 6 and also in chapter 7, we focus more on kidney fibrosis, and also we tested a possible drug candidate for this application. And finally, in chapter 8 and also in chapter 9, we focus more on stem cells. And in chapter 9, we also started to develop the first steps toward the production of a more complex kidney fibrotic model. In our research, we studied the urinary system, which, as we all know, the kidney is the main organ. The functional unit of the kidney is known as nephron, which has a very precise functionality in terms of absorption, secretion, and reabsorption of nutrients, molecules, water, and ions. In particular, we studied the first segment of the nephron called the proximal tubule, and also we focused on the interstitial part around this, and this overall part is called the tubular interstitium. Kidney fibrosis is one of the multiple pathological endpoints of chronic kidney disease. However, it's not fully understood yet. And fibrosis can actually affect multiple compartments of the kidney, and according to which compartment of the kidney fibrosis is affecting, then we can actually have a different terminology. For our applications, we mainly focus on the interstitial fibrosis, which is actually an increased deposition of extracellular matrix, ECM, between tubules and peritubular capillaries. And of course, this also affects the stiffness of the tissue. In a preclinical stage, we can actually use two different types of models to study kidney fibrosis, as animal models, which are known as in vivo models, and also laboratory models known as in vitro. Normally, nowadays, regarding in vivo models, the most used animal is the mouse. And according to which surgery the mouse undergoes, then we can actually have a different type of in vivo model. For example, one of the most common one is based on the cut of the ureter, and this really induces a local fibrotic environment in one of the two kidneys. However, we mainly focused on in vitro models. And once we talk about in vitro models, we actually have multiple type of models, starting from simple to the cell cultures, meaning that the cells are actually cultured in a plastic surface in 2D, for example. And also we can further increase the complexity of these models by including, for example, membranes or hydrogels. Of course, we can also culture the cells in 3D, and we can actually create clusters, which are known as, for example, hydrospheroids or organoids. And we can even further increase the complexity of these models by using, for example, on-chip models, also biofabrication technology, and also a hybrid approach where we combine multiple technologies. For our models, we mainly focus on biofabrication techniques, and in particular on bioprinting, which can actually explain as the production of complex biologic products from raw materials, such as living cells, matrices, and molecules. In particular, in our thesis, we focus on two main different techniques. The first one is called the microfluidic bioprinting, and the idea is that we can actually bioprint filaments that have a core shell arrangement, meaning that we can have actually a filament that has multiple concentric layers, where the inner part is called the core, where we place the cell compartment. Then in the shell part, we actually place the polymer, and the shell actually is the outer layer. However, we also use the pressure-assisted printing technique, or simply called extrusion-based printing. And for our applications, we were not bioprinting any cells directly. However, we were printing sacrificial hydrogel, which could actually make for the production of channels in our models. From chapter 4 onwards, we focus more on the proof of concept. So in this chapter, our aim was to really try to mimic the proximal tubule and also the intricate exchange with the vasculature side. We used the microfluidic bioprinting technique, and our goal was to print filaments. These filaments actually could mimic the architecture of the kidney. In particular, in this panel on the left, we have in a green color the proximal tubule-like structure, and then in the blue color the endothelial capillary side. And we showed that we were able to have a very precise deposition of cells, and this could actually mimic certain aspects of the architecture of the kidney. Then we use an extrusion-based system in chapter 5. Once again, we started from mimicking the proximal tube and also the capillary side by 3D printing this 3D printer single-channel system. And also, we focus on the tubular interstitium. So we were able to print two channels with a gel compartment in the middle, and this overall structure actually can mimic the tubular interstitium. Also, in this slide, we showed some results that we obtained. In the first panel, top left, we were able to mimic the proximal tubule, while in the bottom left, the capillary side. And finally, on the right, we have the overall structure, which was fully covered by cells. In chapter 6, we aim to try to mimic the kidney fibrotic environment in vitro. In a healthy-like situation, we have a nice epithelial cell layer, as shown, and also we have the fibroblast cells in the extracellular matrix. However, an injury can actually trigger the formation of fibrosis, and then multiple phenomena can take place simultaneously, as, for example, the death of epithelial cells and also the over-secretion of proteins and ECM. We created the following in vitro model, which actually is made by a perfusion chamber and also by a bioprinting technique, which we could actually mimic the tubular interstitium and also induce a fibrotic-like environment. This is one of the results that we obtained for this study. On the left, we are showing our bioprinted model, and by means of histology and Picoserus red, which is a specific stains that stains all the collagens in our model, we were actually able to prove that the fibrotic-like model was actually having an increased deposition of proteins, of collagens, and this is one of the main features of kidney fibrosis. So this was a very first good indication that our model could mimic certain aspects of fibrosis. Then we compared our bioprinter model on the left toward the fresh human kidney cortex tissue on the right. And again, by histology, we showed a similarity in terms of secretion of proteins and collagens, as shown by the protein gradient being stained. In chapter 7, we tested a possible drug candidate called pirfenidone. This drug is actually FDA approved for the treatment of multiple fibrotic tissues as cardiac and also liver. However, for the kidney, we don't really understand yet how this drug works. So we thought about to actually work more on this aspect to actually show, to actually try to understand if our model could actually give us some insights in this. We did multiple studies regarding this aspect, and in particular, we used our model to first induce a fibrotic-like environment, and then we actually tested our drug compound in the model. The results regarding the drug compound are actually shown in the orange bar in these graphs, and in particular for the collagen 1 and 3 measurement from sections, and also regarding the total protein concentration and also the ROS measurement, we actually were able to show an inhibition process from the drug. So this can be a very first indication that this drug can also have an inhibitor effect regarding the progression of fibrosis. In the last two chapters, chapter 8 and 9, we focus more on stem cells. Nowadays, stem cells are a very hot topic in our field for our applications, and these stem cells were actually cultured in cluster to form organoids. An organoid is a 3D cell cluster, and in a very simplistic way can actually be explained as a mini organ. So once we have organoids, these organoids can mimic certain aspects of the organ morphogenesis and also functionality. However, these organoids are nowadays, we cannot really create organs in the labs yet. For example, some of these limitations are maturation, vascularization, reproducibility, and also scalability. Our goal was to try to tackle some of these limitations. So, for example, by means of a bioprinting technique, we started from bioprinting single cells as MM and UB. From these single cells, we actually were able to form some clusters within a few days of culture. And afterwards, this cluster became organoids after 2 weeks of culture. This is a video actually showing how we were able to start from single cells in the first picture top left, and then after a few days of culture we were actually able to obtain our clusters in culture in a fully automated way. Finally, in the last chapter, we tried to further increase the complexity of our organoids. As I said, also the maturation and also the vascularization are very important limitation of our organoids and also the organoids in the publications. And our goal was to really try to combine the organoids with the chip from chapter 6, and also adding endothelial cells in the model, and this combination actually showed us that we were able to increase the vascularization and also to create simple vasculature. And we believe that these can be really the first steps toward the production of more complex fibrotic-like models in vitro. To conclude, we started from reviewing multiple papers and publications. Then, from chapter 4 onwards, we started working in the lab to really try to create 2 different proof of concepts. In chapter 6 and in chapter 7, we focused more on kidney fibrosis and also testing a possible drug candidate. And finally, in chapter 8 and also in chapter 9, we worked on stem cells to also try to create a new in vitro model, including 3D printed chips and organoids. Finally, I would like to thank my supervisors, Dr. Carlos Mota, Professor Enzo Moroni, the entire Motas group, CTR group, Molin Institute, our collaborators in Germany, in Aachen, and also in Milano, in Italy. Thank you all.
Thank you for this very clear presentation, beautifully illustrated with images. Your supervisory team has selected a team of experts that first of all have assessed the quality of your thesis and which was approved. All these experts are now present here, and they will want to ask some questions that have remained. So that is why we will start the opposition now. The opposition will be now started by Dr. Truckenmüller. He was the supervisor, the chair of your assessment committee, and he has the expertise in micro tissue engineering at the Merlin Institute at this university. Please go ahead.
Thank you very much, Prorector. Dear candidate, please accept my congratulations for your thesis, which I very much enjoyed reading. I think it was really in the sweet spot of bioengineering, so engineering work, materials, biology, and everything conducted very well, and also quite an extensive booklet here. I would also like to extend my congratulations, of course, to the supervisory team. And now let us go to the questions and the discussion. So my first question concerns Proposition 9, where I would be grateful if one of the esteemed paronyms could read this proposition for us.
We tend to avoid failure at all costs, but our smarter missteps are worthwhile because they can force us to take a different path that point us towards personal and professional success.
Thank you, Paronyms. Dear candidate, may I ask what was your smarter misstep, potential misstep, and the different path you potentially took? And obviously, you have been successful according to the booklet. So what was your potential misstep?
Dear highly esteemed opponent, thank you for your words and also for the question. Indeed, this thesis has been a teamwork over many years. And so really here we have to thank so many people from the supervisor, me, my family, and also the people here online and on site. Regarding your question, in particular, actually, yes. So if I look back actually to my journey regarding first the master internship and then the PhD over these 4 or 5 years at Morlin, I can really recall so many mistakes. Honestly, I'm not afraid to say this. I'm very proud of every single mistake that I've made. I never cut myself in the labs so far, at least. But yes, so regarding actually planning experiments, of course, I've made some mistakes. But then actually from these mistakes, we also learned a lot. To actually really answer your question in particular, right now, I can actually think of multiple errors. Maybe one of the most simple mistakes that I made really when I started is that regarding the printing technique. So sometimes we really see a printer and we say, oh, that's easy, right? You can actually press a button and just it works. So sometimes really I underestimate how long it takes to have an in vitro model out of the printing or bioprinting process. So normally it takes weeks if you're lucky, but also normally months. So something that I learned is that really never give up, because it takes time. So and then actually spend time on the printer, because also this thesis was fully based on printing and bioprinting techniques.
Thank you for these insights. Candidate, my next question would actually concern chapter 4. There you describe that you use a pressure-driven microfluidic bioprinter, but you talk also from studies which used a flow derived system, for example, using syringe pumps. Can you give us some ideas about the pros and cons, maybe particularly in conjunction with what you worked on with that kidney project? Why was that what you used, probably the system to be preferred?
Yes, also this is a very good question. So that chapter was published in 2020, and back in time, actually, this microfluidic bioprinting technique was quite recent. If I recall correctly, our paper was really the second one published regarding this technique worldwide. So if we actually think about the kidney architecture, and in particular the nephron, we can actually think of this architecture as a sort of multiple concentric layers, right? So for example, we have like this proximal tubule, and then actually around we can actually have the vasculature side. So if we think of the nephron from this technical engineering point of view, we actually thought that the microfluidic bioprinting system, and in particular using a core shell printed, could actually be very good options. In that chapter in particular, we did not use any pump system or any perfusion indeed, because our goal was to really try to prove our point, so to really try to show that we could use that system for actually making the nephron-like structure. And then from this first proof of concept, in chapter 6, we developed then a follow-up study where we used the same technique, however changing the polymeric solution, the bioinks. And from this, we were actually able to make also perfusion in our model by using a rocking shaker.
May I very briefly, candidate? So I was mainly interested concerning the printing, so not so much the perfusion of the built model. What would be what you understand now, with your current understanding, be the pros and cons of pressure or flow control on the printing?
Sorry, I misunderstood your question then, yes. So if we think about the microfluidic bioprinting system, this one can actually provide many advantages compared to other bioprinting systems. So for example, indeed it's a pressure-based system, so we are talking about very low pressures compared to other systems as extrusion-based standard systems. So we're talking of the millibar range, so it's very low. And also we are able to extrude a low viscous solution, because in our system the viscosity really plays a very important role. So the combination of low pressures, of low viscosity, of low concentration hydrogels could actually allow us to create the in vitro model, if I'm answering your question.
I would go to, stay with chapter 4 and go to chapter 5. In both cases, you mentioned that it's challenging to impossible to use other technology which is very popular for preparing these miniaturized models. So classical micro technologies relying on lithography. And there, in discussing or stating that is just challenging to have these curvature introduced. Maybe after compiling and writing the thesis, maybe you know, meanwhile, a little bit more, or you knew already at that time, are there some possibilities, and what would that be to make, for example, at least semi-spherical perfusible lumen?
Yes, so if we are actually talking about semi-circular structures, then also in Netherlands there are several companies making actually these chips, so these are commercially available. However, actually, these chips present many advantages, as for example, they are really compact, so in a well plate, so in a very small environment, you can actually have multiple chips with multiple channels, and this can actually really also allow you to test multiple conditions in the same setup. So this also means speeding up the process.
And if I may, do you know how they are made?
Yes, by soft lithography mainly, but again, if
I may, how is the master made?
On that, honestly, I'm not really sure. So, of course, since these are commercially available, it's always tricky to. Yeah.
If I may, still a last question. I try to be fast. My last question would concern chapter 5. And there is actually, I'm interested there to understand, so the importance of curvature is highlighted there. And so we see, I think it's in figure 2, some nice cross-section, very, very well made, very well engineered. But do you have some insights how at the interface to that, from the tubal or the capillary to that interstitial compartment, was that roundness also preserved there?
Yes.
Did you have some imaging allowing you maybe to assess this a little bit, and do you know how that looked there?
So, being honest, I don't think we ever took pictures of that specific part in the chip, but also, of course, we have the central compartment in contact with these hollow channels. So, yes, it's something that we might have to look further for sure. Thank you for the point. Yes.
Thank you, candidate. Dear Prorector, I'm very much satisfied with the answers of the candidate. I have no further question, would give back the procedure for further handling to you. Thank you.
Thank you, Professor Truckenmüller. The opposition will be continued by Professor Koolman. Professor Koolman's expertise is in chronic renal failure, and he's working at the Department of Internal Medicine and Division of Nephrology at the Maastricht University Medical Center. Professor Koolman, go ahead.
Thank you, Mr. Prorector. Dear candidate, I also want to compliment you and your supervisory team on this beautiful thesis, where you really took a very stepwise and structured approach into a difficult topic. And I would like to invite your other paranymph to read proposition number 10.
If you can think of it, you can bioprint it.
Thank you. So that's quite a bold statement. And but if I went through your thesis, I saw a lot of very interesting and beautiful results. But the conclusions and the valorization I found were a little modest in a way. I think studying kidney fibrosis is a laudable goal. It's very important. But we also, I think the topic also lends itself for dreaming on a little bit. And the only place where you did this was actually in chapter 8, where you're talking about the foundation for future kidney disease treatment. And I would like to discuss a little bit with you on your thoughts about it and take a kind of a structured approach there. If we look at kidney, let's say the treatment of patients with kidney failure, we have kidney replacement, in a way replacement of kidney function by means of dialysis therapy, and we have transplantation. And this is, both have their challenges, as you know. And the first thing I would like to discuss with you, if, let's say, the tubular cells which are going to bioprint would lend themselves actually on an industrial scale in the near future to as an add-on to dialysis therapy, because there has been a kind of many developments to add tubular functions to the dialysis treatment itself. Would, let's say, bioprinting in the near intermediate future be possible on an industrial scale really to serve as a source for these kind of add-on treatments? That's my first question.
Dear highly esteemed opponent, thank you for your question and also the kind words. Regarding actually your question, indeed, we have really so much hope in this technique in bioprinting. From what I've seen in these years, to actually move a concept from academia to industry to companies is not as easy as you might think or as someone might think. Of course, sometimes the aim of academia and companies can be different. If we think about companies also in this region, in Limburg, actually there are some companies, also very big companies, international companies, that are really trying now to know more about this technique of bioprinting. So I really hope that the transition to also see bioprinting more spread out also into companies can be happening really soon. So I really see the potential of the bioprinting. I don't agree when people say that bioprinting is a new technique, actually, because it has been around for decades now. But I think it's really time that also companies really invest more on this, because the potential are endless from this modeling to drug testing and much more. And also regarding dialysis, it's something that we never thought about. Maybe it's a bit farther from the aims of our thesis, but of course it's a very good point that we can also think about in the future.
Could you really, without going into the details of connecting into a dialysis device, could it be used actually your techniques in the future to create a lot of let's say numerous cells actually in an environment which could, let's say, where they could live for, let's say, some days and be functional?
Yes, if I'm not mistaken, I think in Twente a group is really working on the dialysis and on these applications. If I got your question correctly, you actually asked me how to combine dialysis and bioprinting.
No, it's not actually not the technical details about dialysis, but more actually, can you produce, let's say, tubular cells from iPSCs in a bioprinted environment to make them viable in sufficient ways that you can have a biologically meaningful effect?
Yes, so if we're talking about tubular cells as epithelial cells, yes, so these are called the tubuloids. And yes, the bioprinting actually, as we also showed in one of our chapters, can be really an automated way to start from single cells to actually make these structures in 3D in a fully automated way. And if this would really scale up, as we think it's a very interesting opportunity, then actually, yes.
And okay, that's a clear answer, and I hope you're right. It would be wonderful. If I go then to page 221 on chapter 8, there I saw, and I think that's a wonderful result, that you were able actually to produce organoids with glomerular type cells and to connect them with 2 different types of tubular cells. Is that correct?
So from page
221.
20. Yeah, in figure 1, actually, no, it's only one cell type.
Only one cell type.
Yes,
okay.
and then actually some pages afterwards, in figure 3 of that chapter, we have the combination of these 2 different cell types.
Okay, so that would be a first step together, I think, with the vascularization in chapter 9.
Yes.
Maybe to build a kind of a structure which resembles basically a nephron. Of course, a nephron is much more complicated. It has, as you already also shows, more than 20 cells who need to be combined in a certain way. Do you think that in the future, actually, still the combination, these are early steps, could be expanded in a way that it could also make in itself a meaningful contribution to organ replacement? I don't say that you will be able to mimic, to completely rebuild a new kidney, but could it actually be used as a kind of an add-on, such an organoid, to current organ replacement techniques?
I really think so. I really hope so as well. The question is when, so not if. So maybe like in a few decades or maybe a bit longer, maybe yes. So stem cells really can provide all the cell types in the kidney. Ideally, of course, now we're not there yet. We have so many issues when culturing stem cells in the lab nowadays. So for example, a protocol can work in a lab, and then if we use the same protocol, no, and then we don't really understand why. So once we actually culture stem cells for the first time, this really means also starting from like really the basics to optimize the protocols, and as we did in that chapter, and then from that point to actually increase the complexity.
Okay, so but you're really talking about decades then. It's not because if we look at the Dutch Kidney Foundation, they are much more, let's say, positive in a way, and say, okay, we'll probably be there around 2035, 2040. Is that not something, because that's already in 10 years, is that not something which you believe in? Would it be too optimistic, you think?
As I said, I made plenty of mistakes in these years. Maybe in 10 years, I don't think so, honestly. If you ask me personally, maybe it will take a bit longer because also the kidney is very complicated. Probably is one of the most complex organs. If I ask you how many cells a kidney has, we don't have an answer, for example. People can argue more than 20. People can say only to more than 20 epithelial cell types. We don't know so much on the kidney yet, so it's still very complicated.
But do you still think that you will need all these cell types to have a kind of a functional kidney? Do we really need to rebuild, you think, a kidney as nature made it, or could it also be made in a little bit simpler way and still be functional?
If you ask me at this moment, I think indeed the best approach is the combination of multiple techniques as bioprinting, biofabrication, and also stem cells, organoids, as we did in these 2 chapters. Because at this moment our differentiation protocols and also culture mediums cannot really provide actually. It's hard to actually have all the cell population of the kidney in our organoids at this moment.
Okay, thank you for your very clear answers, also very realistic answers. I'm very happy with your answers and would like to give back the word to the pro-rector.
Thank you, Professor Koolman. The opposition will be continued by Dr. Janssen. Dr. Janssen is a group leader at the University Hospital in Aachen, and her expertise is in nephrology and hypertensive diseases. Dr. Janssen.
Thank you, Pro-Rector. Dear candidate, I would like to congratulate you with your beautiful work. There was an impressive amount of the chapters that you accomplished. I would also like to congratulate your supervision team. And I read the thesis with great interest, and maybe you're not surprised that I would like to start off with the organoids that you have also used in this work. I would like to start with chapter 9, and I would like to, I'm curious to hear about your ideas about using organoids into chips, and especially if you think of the context of fibrosis and towards developing anti-fibrotic drugs, wouldn't this be way too complex, and why don't we just simply use your other beautiful model where you use the fibroblasts and the epithelial cells and use that for drug screening? Or do you think we definitely need the organoids when in the context of anti-fibrotic drug development? So I'm curious to hear your thoughts about this.
Dear esteemed opponent, thank you for your questions. Indeed, these are all very good points. And of course, answering to these questions is tricky. I'm sure that multiple experts will have multiple opinions. Regarding myself, actually, I can say that primary human cells in particular still provide, I think, at this very moment, the best the best culture protocols. So one of the main issues that we have in the labs, as I was saying before, is really these differentiation protocols. However, if we only focus on primary human cells, for example, then we cannot really combine all the multiple cell types in the kidney or in any other organ, right? So, for example, we have so many technical limitations. So, for example, just simply which culture medium to use with so many different types of cells. So, if we think about this technical limitation, then we can actually really work on stem cells, on iPSCs. If I also think about the multiple groups at Morlin, for example, and also outside of Morlin, many different groups are really working on this topic nowadays. So many different groups really see the potential of this. I also agree on this, and also if I would really continue this work, I would only work on stem cells. It's very difficult. You might have to combine indeed the organoids with, for example, other platforms. Can be organ on chip, can be biofabrication, bioprinting, but indeed I really think it's our best approach.
Okay, thank you. Then I would like to move to figure 4, where we see the double-layered channel with the UVEC cells and then with the organoids in the middle, right?
Yes.
So you cultured the UVECs first for 7 days, if I recall correctly. And in previous chapters, I saw this very nice CD31 expression, but here I see no CD31 expression. What happened with these UVECs? Don't they like this chip, or did you have a new batch of antibody, or what happened here? Why you don't see any CD31 expression?
Yes, so you're mentioning figure 4, you said?
Yeah, figure 4, chapter page 250.
Indeed.
Yeah.
Perfect. Yes, so indeed in this specific chapter, we culture for 1 week only UVECs in their AGM2 medium, so in their native medium. We all know that the UVECs are actually very sensitive to the change of medium, so if you culture them with a different medium type, then the UVECs don't really enjoy this. However, from our data in this chapter, we showed that first the pressed blue from figure 2, I think, actually showed us, yes, from figure 2, that also once we culture the UVECs in the advanced L' medium, actually these cells still show the CD31 staining, but as you said, also the morphology is different. Yeah. So that's a very good point, but we were still able to measure the CD31 staining. So we think, and also this data suggests that the cells are still endothelial cells, and from what actually we saw in this very specific figure 4, and also in other studies that I was doing really until last week in the lab, indeed the morphology changes, but the endothelial cells are still in the chip even after the first week of culture, once we change the culture medium to advanced L', and the staining of endothelial cells is there, this CD31, and also the branching from the organoids, we think these can be capillary-like structures. So from our Z-stack images, they also showed to be quite hollow channels.
Okay, thank you. So I was also wondering, so we know from the organoids that they don't contain the correct endothelial cell niche. So the progenitors in there are not, if you compare them to fetal kidney, are not comparable. Let's say there's a different niche, so to say. So there are some groups that have shown that certain transcription factors have to be activated to get the correct endothelial niche. So do you think that so this UVAC, let's say chip, so to say, that make the connection to the organoid, will that stimulate the correct endothelial cell niche progenitor differentiation, or do we definitely different endothelial cells in the organoid? So how can we now make, basically we want to have vascularized glomerulus, right? That's the first step, and the peritubular capillaries. Are we in the right direction by simply, so to say, adding this organoid with UVACs and good to go? Or what should we do now to make this really working? What is your idea?
Yes, so if we... first of all, at this stage, we cannot really answer this question if the organoid is able to actually create its own vasculature once it's placed in the chip, or maybe actually the UVACs from the chip that actually go into the organoids to actually vascularize the organoid. So this is a question that at this moment we cannot really answer, and we don't have data, unfortunately, to answer this question. However, I still think that the vascularization and the maturation of these organoids is very limited. So if we don't have a protocol in the lab that actually really boosts the vascularization of these organoids, then the only option is to actually combine multiple approaches, as we did. Also, if I think about publications and also how the multiple groups from Japan, US, and also Europe work on this topic in these last 5-10 years, I think they always combine organoids and UVACs and endothelial cells and biofabrication or on-chip models. So I think that from this point of view, we are in line with the other groups, which can be good or bad sometimes, but still I think it's our best approach.
Okay, thank you. I'm also not sure if the UVACs are the best, because these are macrovascular cells, right?
Fair point, yeah.
And I think also there other endothelial cells are available, so it might be good to brainstorm about what would be, let's say, the next level of cell models to be used.
I fully agree on that, yeah.
And then I would like to move to the chapter about the microfluidics, where you also printed, let's say, the organoids in the microfluidic setting, and there you show this albumin uptake. But to me, so this was, I think, in chapter 8, right?
Yes.
And then we go to, I think it was figure 4, if I recall correctly. So there we see albumin in the figure, but actually I was wondering, like, okay, we see this orange, yellowish dot, let's say, but is this really albumin active uptake, or how would you study if this is really uptake, or it could also be simply cleavage to the, what is it, podocytes, I think, where we see it mostly. Actually, you would expect it to have it in the proximal tubule, right? Mostly, if you have cubolin and megalin expression, of course.
Yes.
So how can you convince me that we have albumin uptake endocytotically related?
I think indeed one of the most hard part of our jobs is to try to make a point and to try to convince the committee in this case, or the community that is working on the similar topics. Indeed, I agree with you that this picture maybe is not the most clear one, but it's something that we might have to still work on this. The nephrotoxicity assay that we also did showed some functionality of our organoids that were actually able to be responsive to the nephrotoxicant. But regarding the albumin uptake assay for the bioprinted organoid, it's something that we might have to work more on this. Yes.
Okay, happy to brainstorm about it.
Yes, please.
I'm satisfied with your question, or with your answers. Thank you very much.
Thank
I'd
you.
like to give the word back to the pro-rector. Thank you.
Thank you, Dr. Janssen. Your position will be continued by Dr. Mihaela. Dr. Mihaela is assistant professor in pharmacology at the University of Utrecht. Welcome to Maastricht.
Thank you very much for the kind welcoming, and congratulations to the candidate for this impressive work. As I was reading, I came with a lot of questions, of course, and before I do this interrogatory, friendly one, I would like to ask one of the paranims to read statement number 4.
The synergy between technology, biology, and biomaterial has to be concurrently investigated.
Okay, thank you very much. Okay, so let's start. I come from a group of pharmacology, so most of my questions are going to be related with drug testing, but I want to put it in the context of the technology that you have developed, the biology and the application for drug testings. So I would like to start a little bit more general. So in your opinion, what characteristics would define an ideal in vitro model for drug testing? You have already touched upon in chapter 5 and in chapter 8. But I would like to have your opinion on that and to give you a little bit of hint on which direction I want you to go. What should you prioritize in such a model, the functional replication or the structural accuracy?
Dear esteemed opponent, thank you for these questions. Yes, that is a very good point. So if I would actually really think about in vitro models, then actually I would even do a step before that. So I would really focus on a technique that is able to make these in vitro systems, models really fast, in a fast way, and also in a cheap way. Because sometimes we really underestimate these aspects, and that's also something that we tackle in chapter 5, for example, with the second proof of concept, chapter 5, yes. And yes, so if then these aspects are taken into account to then really answer your question, I think that nowadays the architecture of the in vitro model really plays a big role. So, of course, by using this technology in particular, we also had some technical limitations. So, for example, even the size and the diameter of our channels. But we were actually able to obtain these models in a very fast, cheap way, and also with these very nice circular cross-sections. So I would say architecture.
Yes, so you would consider that there are these trade-offs on between the physiological relevance and the practical utility and in the designing of your in vitro models. But I would like to challenge the vice versa. So if it doesn't look like a proximal tubule, does it mean that it's not a proximal tubule as long as it behaves like one?
I mean, in a very simple term, like if we make like an engineered channel, then we can actually use that channel for many tissues, and the application in our body can be like a capillary, can be, for example, a proximal tubule, can be anything, right? But of course, the cell type makes a big role, and also, I guess, for example, also the shape of the channel itself can also play a big role for sure.
Okay. So let's now go into the experimental part and see how your answers will fit into what you have presenting here. So in chapter 4, you present the core shell format where you have the endothelial cells wrapping a channel that is filled with proximal tubule cells. And then eventually, at the end of the culture time, these proximal tubule cells create a lumen. And in the readouts, you focus a lot on, okay, how the cells, if the cells are alive, on the metabolic activity. Can we say we have a functional proximal tubule now?
So the chapter 4, as you mentioned, was really our first step. Personally, it's my favorite chapter in the thesis for many reasons. But to really answer your question, that channel, that proximal tubule was not as complex as a real proximal tubule in the human body, but again, we showed some evidence to be able to mimic certain aspects of the proximal tubule in the body.
Yeah. So then you move to chapter 5,
yes.
where you try to do this organization on also on the tubular part.
Yes.
And you mentioned that this system can be used for drug testing. But you haven't done any drug testing in chapter 5. What do you think? Do you think you didn't do it because you think the model is not ready for drug testing, or it will be in a different chapter? And what do you think the model still needs in terms of it to become suitable for drug testing?
Yes. So if we're talking about chapter 5, the drug testing indeed was not there. We focus on drug testing in chapter 7, but again, switching to the microfluidic bioprinting system. Something that I learned really in the lab is that doing drug testing in vitro nowadays seems really easy, but we have to really understand and answer so many questions. So, for example, how to even provide the drug to our in vitro model. Also the concentration, also, for example, for how long. So it's really tricky, right? Yeah, so to answer your question, if we would actually want to do some drug testing in chapter 5 model, we could do it, I believe, and yes, I think so. But again, we have also to really understand also which cell types to use. So, for example, if we are using for chapter 5 the proximal tubular cells and again the UVEX as we did, then we also have to understand a bit of the drug and how it's working, the mechanism behind. And again, not always we have these answers in papers or in literature.
Yeah, and then you also mentioned that there are many other models that are similar to yours, also double channel, gel in between, also another interstitium. So I was wondering, if you are going to do a drug testing, would you look, for instance, in the concentrations that give you a certain response, let's say cytotoxicity? Would you compare your findings with other in vitro models? Or, and here I would like for you to fill in, if there is any other reference that you might use?
Yes, so for example, in chapter 7, we really tested this drug. And as you asked me to try to understand which concentration of this drug to use was not easy. So our only reference regarding in vitro models was a few papers in 2D. So then the question is, if the drug works in 2D, it will also work in 3D in our model.
Yes, but what I want to ask is more into the, okay, in your model, you get a IC50 of 10 micromolar. Yes. And in a competitor model, you get 50 micromolar. And in Utrecht, we do the same with our model, and it's 100 micromolar.
That's an issue.
Which one do we get as a reference? Which of these models is going to be the one that will move forward? Or do we need to look at other kind of standards? And here I want to kind of challenge you. Where should we look for references into what other people are doing? Or
yes, so indeed, sometimes publication can be misleading, unfortunately. Like multiple groups can do something different, and that difference can be a big difference sometimes. So in our case, we chose a drug concentration based on paper, but also we had a chat with some pathologists in San Diego, in California, about that. But then to really try to answer your question, we have to try in our labs as well. So, for example, if I would see 3 different papers with 3 different IC50, I would really try every single concentration with our own cells in our own setup. So I would really do like a force screening on my own.
And this will be my last question, so also I would like a brief answer. So I'm also looking where I wanted to also go a little bit with you as what is the available data in terms of clinical practice, because we already have some information there from these drugs. And should our in vitro models match this information, for instance, nephrotoxicity in this case, that is in clinical, and what do you think should be the readouts that will match in vitro and in vivo? Are there these readouts that can be translated from in vitro to in vivo?
So based on my own experience and from what I've seen in my research, the data from preclinical and clinical regarding drug testing does not match for multiple reasons. So for example, so just simply a human body, so a clinical stage testing is much more complicated at this stage compared to an in vitro model. So then, of course, we have to make a choice regarding in vitro. I would love to really see very soon in vitro models to be as complex as certain aspects of the human body tissues, but at this moment we are not there yet. So then again, it's really a matter of trying in the lab and see how it goes. And this is part of doing research, doing mistakes mistakes no, but just learning more how it works.
So we hope that in 4 years another Gabriele will be here and tell us more about this.
I hope so, yeah.
Thank you, Dr. Mihaela. The opposition will be continued and finalized also by Dr. Sinha. Dr. Sinha is present online and will appear on the screen. Now, his expertise is in biofabrication at the Region Biomedical Company. Dr. Sinha, go ahead.
Thank you, Prorector. Dear candidate, I would also like to congratulate you and your supervisors on this very nice piece of work. It was a pleasure reading it, and I really enjoyed that you stuck to a core set of technologies and slowly built up the complexity of the models from the earlier chapters towards the later chapters. What I also really liked was that you emphasized on the automation and cost aspects of building these models. And as you know, in my current role, that is something of interest for me. So I would like to probe you on those aspects. So when you say that you built a model that is highly automation compatible, I want to probe you from a perspective of a CRO that would like to do drug screening using your models. What would you like them to do? Build the model themselves, or do you foresee building the model and shipping it to them with the cells in there, or with the channels and the cells and the compounds? Can you elaborate on those aspects?
Dear esteemed opponent, thank you for your question. The audio was a bit unclear, so I hope that I got your question correctly. So you asked me about the automation of the system and also regarding the drug testing, if I got it correctly. Yes. So indeed, once we actually culture cells in the labs, it's nowadays, being fully honest, is a process that it fully depends on the user. And there are better users compared to others ones in certain techniques. I've seen this really also in my previous group of research when I was working here in Maastricht. And but yeah, so if we could really work on an automated process, can be bioprinting, can be a robotic arm, for example, or a similar system, then I think that we can really limit the variability among the multiple users. And once actually we have this sorted out, then really the possibilities are endless. Just to think about, we can make organoids with the same shape, with the same number of cells, with the same functionality, and we can really scale up these for multiple applications, as for example drug testing. And yeah, so I would really push more on this automation. And of course, I know it's challenging. I know it takes time, effort, money again. But yes, it's something that I think is very important.
Thank you. I would repeat a part of the question again, because I think you covered other aspects, but this part you missed, maybe because of the missing audio. But my question mainly was, which aspects of the automation do you foresee the people using the model doing? Would someone make the model, put the cells in and give it to the testers, or the testers can do everything in their facility because everything is automated?
Yes, so if I could actually say what I think about this point, I think the best approach is to have the entire line fully automated, so really from the model to the cell enclosure. But again, I think I can only think about a few groups of research worldwide doing this, or at least they tried this. I think that nowadays it's not a fully automated process yet. So for example, we have to focus more on the model, and then for example, we have to insert the cell in a more manual way. But for example, I also know that Regen is doing a very nice work on this regard. And the goal, I have no idea if this goal has been achieved yet or not, is to really have a fully automated process. So this means ideally to have also multiple modules. For example, one focusing more on the manufacturing and one more on the cell part, and then actually combining them together, for example. But I fully understand that this comes with many difficulties.
Thank you. Then I would ask you about one of the engineering choices that you made that could, in my understanding, become a barrier to the full automation. You picked PDMS. PDMS is really viscous, hard to handle using automated liquid handlers, hard to sterilize. Why did you make that choice? And if you were to move this process to a fully automated system, how would you change it?
So yes, again, the audio was a bit unclear, but regarding the PDMS, yes. So regarding the PDMS, indeed, so what we actually did was first depositing a first layer of PDMS manually, and then actually the printing of Pluronic took place in a fully automated way, and then we also did a second casting of PDMS. So the process was not fully automated. Of course, we can also think about a fully automated process, but then I also wonder if then PDMS is really our best approach for that. So PDMS is just plastic. I fully understand why people use it. I also use that indeed. But then if we really aim to have a fully automated process and also we care more about the functionality of our results, then I would switch to protein, for example, as we tried to do, for example, in chapter 8 when we bioprinted the organoids, the single cells creating organoids. So again, it really depends on the application, and also yes. So yeah.
All right, thank you. Then I would like to ask you on the aspects of the mechanical stimuli that you introduced in your models. So I saw that some of the models included flow. What I found missing in your thesis was how these flow regimen were chosen. You didn't compare. Yes, it's time.
Dr. Signa, you can finalize your question and the candidate may briefly answer.
Yes, so I was missing what is physiological flow and what is pathological flow, and because you saw in the presence of flow in your fibrotic model already a pathological response of fibrosis. Do you think that this was because the flow regimen was not well thought about?
Yes, so for our model, indeed, the stiffness played a very big role. So we also played on that aspect by making this doping with vitamin B2. And yes, so once the stiffness changes, also the biological response changes as well.
Thank you, Dr. Signa.
Thank you.
Mr. Addario, the time allotted for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and the quality of your defense. And I request that you and your company await the result of our deliberations and our return in this room.
All right.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and not. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and not. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Mr. Addario. The degree committee here present has assessed the quality of your thesis and the quality of your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Moroni is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor. Professor Moroni.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law, and in conformity with the decision of the committee here present, I hereby confer upon you, Gabriele Addario, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Dear Gabriele, dear Dr. Addario, what a marathon. It seems that we went through several marathons throughout your PhD. You did these marathons with clear aims, despite the difficulty to formulate a research question. Going back in time, we started at first with a master internship at Merlin back in 2019. It has been a while. At the end of your master, you were not sure of what to do, what you wanted to do next. Despite of this, you asked early about a PhD and what it would entail. With some thought, you asked if there would be any options to do a PhD at Merlin, and you have applied to a position I had opened at the time. After offering you the position in one project, you early expressed that there would be an opportunity, if there would be an opportunity, you would like to continue the topic of your master on kidney and bioprinting. This was in the early months of 2020. Yes, that amazing year where we had to face one of the biggest challenges in our lives. Remote work, shifts, virtual meetings, we had experienced it all and survived to tell the story. After 1 month of your PhD, we swapped your position to a kidney project. So you were lucky, so you thought. The challenge was big. With secondments at collaborators in Aachen, I want to thank Professor Peter Bohr, and in Italy with Professor Marco Rasponi, that I take the opportunity also to thank. The opportunities were endless, but COVID was always on the way. The project evolved over the years, with Gabriele gradually mastering new techniques and expanding his knowledge both on technology but also on biology. With these skills and with some help along the way and with team effort, here I also want to like to send a shout to Dr. Chiara Formica, a postdoc alumni from the group. You started to do bioprinting of kidney progenitors to form kidney organoids, which you used in 2 chapters of your thesis. This work partially funded by the Dutch Kidney Foundation that I also take the opportunity to thank. After establishing all the models, Gabriele embarked on a journey to emulate kidney fibrosis. At first, the mission seemed impossible, but Gabriele was not afraid of the unknown. He started by trying to understand the mission and then on the design of the experiments, which would allow him to achieve the model where he could investigate the first steps of emulating fibrosis in vitro. He was also able to revert the process by testing compounds that were still not applicable, particularly to kidney fibrosis. Gabriele had other missions. The ones that he was most fearful was to go to the lower floors of the faculty building. To mention a few, the collection of primary tissue was something that he was always amazed, to put it in a mild tone. But until the very last days, he was amazed with the lowest floors of faculty building by discovering the new world that was the biobank at the time of eternally storing his samples. From the lowest floors to the greatest achievements, Gabriele, you were always fully equipped to explore the world with a set of skills which I'm sure you will use wisely. But Gabriele's PhD is not only about science, but it was also about after-work activities. Gabriele created a great network of friends which were essentially to succeed on his PhD process. You can appreciate the endless acknowledgement sections of the thesis, and I'm sure that some names were not mentioned in there. I recall some activities with Shivesh on gardening, cooking, and many others. You both ended up on being so alike that you even started to dress alike. Talking about good friends, you always mobilize friends for great activities into altruistic initiatives, such as the marathon to support cancer research, to the donation of your gift today of your PhD graduation to the Ronald McDonald House in Maastricht. This reflects well your human side, constantly supporting a great cause. During your master and PhDs in the Netherlands, it was clear that you mature even further in every way, from the technical to the emotional. Your resilience, lucidity, even when challenged in difficult situations, leadership and proactivity resulted in excellent results showing throughout your thesis, and this moment is a proof of this. Your work was evaluated by world references, and achieving the doctorate is a logical step. A path training and essential skills that you will become expert, that you became expert within interdisciplinary requirements known to achieve the models that you presented today. I'm sure that your new research engineer position at UN Eindhoven will bring you back to the engineering roots, but I'm sure that you will address any challenge with great responsibility and maturity. We hope you continue to excel in the future. I also wish to congratulate your family that traveled all the way from Italy here to celebrate this day. I would also like to thank you for raising this young now doctor with strong values, respect and determination, which are essential for his success in life. I wish also to congratulate your better half which was a great support for this and future milestones, I'm sure. So I'll give the floor to Lorenzo a bit.
Caro Gabriele, qualche parola in italiano. Prendo l'occasione per ringraziarti del grande lavoro che hai fatto in questi anni. Sei sempre stato pronto ad ascoltare consigli e critiche, non ti sei mai scoraggiato, al contrario hai sempre trovato l'entusiasmo e l'allegria di portare avanti i tuoi progetti. Queste caratteristiche, assieme alla tua dedizione per il lavoro, fanno di te un esempio per tutti i tuoi colleghi. Gabriele è una persona solare, un amico per tutti, con cui è così facile spaziare da argomenti seri come i nostri progetti di ricerca o l'ambiente ideale per le prossime sfide lavorative, ad argomenti più frivoli come le partite di calcio, anche se tifi la squadra di Milano sbagliata. Un ragazzo, no, ho sbagliato qui in realtà, un uomo con un cuore grande che abbraccia la vita, sani principi e professionalità, tutti i valori di cui voglio anche complimentarmi con la tua famiglia ed estendere anche a loro le mie più sincere congratulazioni per questo importante obiettivo raggiunto. Sai che se avrai bisogno di consiglio o semplicemente di essere ascoltato, sarò sempre per te presente.
Let me also thank Professor Lorenzo Moroni for all the shared supervision efforts and giving the possibility to support and secure the funding necessary for that fourth year of a PhD. You were funded initially by the Marie Curie funding scheme, which didn't allow that, but then Lorenzo made an extra mile to facilitate that. As a final moment, I would like to thank all the members of the committee for taking the time to read critically your thesis and for being with us today. Professor Jeroen Kouwman, Dr. Sylvia Maila, Jitse Johnson, and Dr. Ravi Singh for adjusting your busy schedule and for joining the assessment committee, and the prorector, Professor Ramakers, for sharing the defense today. I would like also to thank Professor Roman Trockelmuller for sharing the reading and assessment committee. Thank you for working together with other committee members to ensure the tight schedule. I'm sure that Gabriele also thanks you for that. Finally, I would like to thank your friends, colleagues, and scientific family for joining in person and online, and Dr. Marco Rasponi and the Synergia project partners, collaborators, and fellow researchers within the consortium for all the support during the second months and throughout your trajectory. Gabriele, Dr. Addario, we finish today with another milestone of both of our lives. We hope we have the opportunity to work together in the future, this time not as your boss, as you would like to call me. Hope you make the best of those engineering and biological tools you have mastered in your new job at the University of Eindhoven. The job hunt was a job on itself, but you were determined and succeeded. It was a pleasure to work with you. Keep in mind, work smart, not hard. Thank you.
Esteemed Dr. Addario, it is my honor to congratulate you on behalf of the University and the Board of Deans with the degree that you have acquired. I also would like, of course, to congratulate your supervisory team, Professor Moroni and Dr. Amota. Of course, also congratulations to your parents and your sister. Good that you came. You could come to Maastricht. Congratulations to your friends, and in particular also to those that have collaborated to your work. Research is a collaborative effort. Many of, and we can see that in the chapters and in the publications that came from your thesis. Many have collaborated with you on this and helped you in a certain way. So also those I would like to congratulate. You have presented us with a beautiful thesis, a lot of work, important work for the future. Hopefully, let's say the basic aspects that you have presented, the technical aspects and the biological aspects, will come to final application in, let's say, for medical care. And I admire the crosstalk between the different disciplines that you have been able to combine in your thesis. With respect to your defense, I think I can say from the deliberations that we had that you replied in a very honest way, humble even to a certain extent, and the answers were also realistic. You didn't exaggerate, and you are apparently and obviously aware of the limitations of your work, which is very important for a scientist. I understand that now you have become a research engineer at the University of Eindhoven, and you will continue the important work that you're doing on biofabrication. And I wish you lots of success in your career and lots of happiness in your life. Before I finalize this ceremony, I have a few announcements to make. I would like to ask the audience to leave the aula and to go to the rafter. After my finishing the ceremony, I would like to ask the parents to stay in the sister to stay in the room, because we will make some pictures here, and I would like to give Dr. Signa the chance to congratulate you online. And after that we will go to the staircase and make another picture, and we will come to the rafter and join you for a drink. Having said this, I now close this ceremony.